Today we will be going over the blood chapter. We will be sharing with you a summary of our notes and a few tidbits to help you remember. Hope this helps. A quick look at the components as they are flowing through your blood vessel. A quick look at the components of blood. Blood is made up of three components. The red blood cell layer, which is just going to have your erythrocytes, that's 45%. Then the white blood cells and platelets, which is only less than 1%, will make up this clear whitish buffy coat. Next is the plasma layer. The plasma layer itself has many other components, which we will discuss later in our slides. And something to note on the physical characteristics, he will want us to know 7.35 to 7.45, slightly basic for the pH of our blood. It will come up multiple times throughout the course, so make sure you remember. The functions of blood. Blood performs distribution. Blood distributes oxygen and hormones throughout the body and also removes carbon dioxide. Regulation. Blood regulates temperature and pH and maintains fluid volume. Protection. Blood clots in order to prevent against blood loss and uses antibodies to prevent against infection. This next slide is pretty self-explanatory. The blood plasma is mostly made of water, 90%, and only 10% has solutes. The solutes of blood plasma are listed as follows. We have proteins. The proteins are made up of albumin, globulin, clotting agents, hormones, and enzymes. It also has nutrients, which are glucose, amino acid, fatty acids, and glycerol. Electrolytes, yes, those things that they say on sports bottles, you have those in your body already. Those are charged ions, anions which are negative, and cations which are positive. And respiratory gases, small amounts of oxygen and large amounts of carbon dioxide. Those are traveling through your blood as they're being distributed throughout your circulatory system. And it also contains non-protein nitrogenous substances like lactic acid and urea. Here we have a closer look at our red blood cells, our erythrocytes. Their structure is a biconcave disc without a nucleus. I remember these because they look kind of like a little lifesaver. And these don't have a lot to them, not a lot of structure. These are essentially little sacs of hemoglobin. And them not having structure actually helps them. They are very bendy and very flexible to allow them where they need to go to squeeze in and out of vessels. They can fold almost completely into a taco shape, all the way up until about 120 days when they become a little rigid, a little old, and usually end up stuck in the liver where they are disposed of. So where do red blood cells come from? They come from a process known as erythropoiesis. It starts out as hematopoiesis, blood cell formation. It occurs in red bone marrow, the axial skeleton girdles, and proximal epiphyses of the humerus and the femur. All red blood cells start out as hemocytoblasts. This process is influenced by the hormonal control of erythropoietin and dietary requirements such as iron, vitamin B, and folic acid. If either of these, the hormonal control or dietary requirements, are not met, clinical problems may occur. We will talk about those next. The clinical problems of red blood cells. Anemia means a reduced oxygen carrying capacity in blood. This can result from a decreased number of red blood cells, which would be hemorrhagic anemia, hemolytic anemia, and aplastic anemia. Decreased hemoglobin levels, which can result in iron deficiency anemia, athlete's anemia, and pernicious anemia. Abnormal hemoglobin, such as sickle cell and thalassemias, or polycythemias, which is a high number of red blood cells. Now let's take a look at the white blood cells. These are our body's warriors, fighting off infection and protecting us from outside invaders. Leukocytes is another name for white blood cells. They are complete cells with a nucleus and organelles. Their main function is immune defense. The types of leukocytes, we have granulocytes, which includes the most popular neutrophils, eosinophils, basophils. We also have agranulocytes, meaning they don't have granules. These include the lymphocytes and monocytes. In our next slide, we'll have an illustration of each. Here we have a slide illustrating the different types of white blood cells. Let's talk about what they do. Monocytes. They perform phagocytosis at the site of chronic infection. Next, lymphocytes contain T cells. T cells attack cells that have been invaded by viruses, like cancerous cells. B cells make antibodies. These antibodies are shaped like Ys and they attach to antigens. Neutrophils have a many-lobed nucleus, making that funky little bubble shape in the center. These perform phagocytosis at the site of acute infection. Next is the red pigmented eosinophil. Eosinophils have large red granules. They kill parasitic worms with enzymes. They do not phagocytize. They do produce antihistamines. This makes them the opposite of basophils. Basophils cause inflammation and they produce histamine, which causes vasodilation. Basophils have large purple granules. Here's a quick look at the stages of leukopoiesis, the development of white blood cells. Now in class, he's not going to go into depth of the growth or development of each white blood cell, but what he does want us to know is each blood cell, whether it be a red or a white, starts out as a hemoblast. But here's a good look so you can kind of see the flow chart of where they come from. Clinical problems of white blood cells. Many of us are familiar with leukemia, but few of us know what it really means. It is when too many white blood cells are produced that are misshapen and non-functional. Other clinical problems of white blood cells are infectious mononucleosis. You remember that, mono from school, the kissing disease, and leukopenia. A small photo of leukopenia is included in the bottom left, and we'll have an illustration next for mononucleosis. Here's just a quick look at the different parts of the body that are affected during mononucleosis. Another one of the major functions of blood is hemostasis. One of the ways that it maintains hemostasis is by platelet plug formation. At the site of damaged endothelium, platelets swell. They become sticky and spiky. Platelets stick to exposed collagen at the damaged endothelium. Chemicals are released. Serotonin, which increases the vascular spasm, ATP, which attracts more platelets. Now you don't want your whole body to clot, right? So to control the size of the clot, prostaglandin gets involved. Here's a quick list of blood clotting phase 1. The formation of Prothrombin activator. Here's a detailed image of the platelet plug formation. It illustrates the trapped red blood cells in the fibrin network that was created by the fibrin mesh, and it also shows some platelets floating along in there. Here's a quick look of the aftermath of a blood clot. Clot retraction and repair is aided by actomyosin protein complex and platelet-derived growth factor. Then, fibrinolysis removes the clot when not needed anymore. Plasminogen, tissue plasminogen activator, and plasmin aid with this. Then we have a control of clot formation. This is powerful and localized. Heparin and antithrombin 3 aid with that. Blood transfusions. This is where our blood types come into play. If a donor and recipient of blood do not have matching blood types, a blood transfusion reaction will occur. This will lead to many other serious complications and potentially death. Here's a short list of clinical problems related to blood. Hemophilia is when a certain clotting factor is lacking. This can result in excessive bleeding. Thrombocytopenia is a low platelet count. Liver dysfunction, not making procoagulants. Prothrombin comes from the liver, and if the liver isn't functioning, it may not produce enough prothrombin. Thromboembolism is when a clot blocks off a blood vessel. We know A&P can be painful, so we wanted to end it with a joke. We hope this short slideshow helped, and we wish you luck in your class.